Today we are going to talk about the mRNA degradation. We have discussed previously about the how transcription takes place, how this mRNA is synthesized, and then it is how do these post-transcriptional changes take place, like capping and polyadenylation, and then removal of the introns and splicing together of the exons. And then we talked about the export of the mRNA from the nucleus into the cytoplasm. We would not discuss how does translation take place because I think almost everyone knows about it, that the ribosomes need to bind to it and then they read the codon. They recruit transfer RNA, which are carrying amino acids, and their amino acids are put together depending upon the mRNA code. So if you remember, we discussed that the regulation of gene expression, it may take place at several different stages, starting all the way from chromatin remodeling, and then transcription, and then post-transcription regulation, and then regulation at the time of translation, and even after the translation, a synthesized protein can even be degraded. So at the moment we will talk about the regulation at an mRNA level, that your mRNA has been synthesized, it has been exported into the nucleus. So either it has already been translated or it's not yet translated. It depends upon the situation, but it needs to be degraded now. So there are multiple factors which would that would determine the lifespan of an mRNA molecule. जी.
सर वो जो आपने इसी mRNA से की ट्रांसलेशन से रिलेटेड है, तो जो आपने अल्टरनेटिव स्प्लाइसिंग में जो बताया था उसमें कुछ ऐसी टाइप्स थी जिसमें हमारा इंट्रॉनिक पार्ट भी मैच्योर mRNA का पार्ट बन जाता है। तो सर जो इंट्रॉनिक सीक्वेंस है उसके जो है उसके स्पेसिफाइड अमीनो एसिड्स होते हैं या नहीं, क्योंकि इंट्रॉनिक सीक्वेंस का तो स्पेसिफाइड अमीनो एसिड्स हम लोगों ने नहीं पढ़े।
नहीं देखें, अमीनो एसिड सीक्वेंस तो डिटरमिन, आई मीन इट्स अ कोडॉन। ये ट्रांसलेट इसलिए नहीं होता क्योंकि इंट्रॉन रिमूव हो जाते हैं। अगर रिमूव नहीं होते हैं तो देखें जैसे म्यूटेशन होती है, कोई एक रॉन्ग, आपका रॉन्ग नहीं, आई मीन 4 में से कोई ना कोई किसी ने एंटर होना है, ठीक है ना। AUGNC में से किसी ने mRNA में एंटर होना है, तो जो हमारा स्पेसिफाइड कोडॉन होता है किसी एक पर्टिकुलर प्रोटीन के लिए वो चेंज हो जाता है। अगर वो वहां पर सीक्वेंस है तो उसको तो mRNA वो सॉरी आपका राइबोसोम जो है वो रीड कर लेगा अनटिल एंड अनलेस हाँ वहां अमीनो एसिड नहीं बनेगा जहां पर वो कोई स्टॉप कोडॉन हो, लेट्स से।
सर मतलब अगर इंट्रॉनिक सीक्वेंस ऐसा हो जहाँ पे वो एक स्पेसिफिक अमीनो एसिड को मतलब स्पेसिफाई ना कर रहा हो तो फिर उसका क्या होगा? सर मतलब वो एक स्पेसिफिक कोडॉन जनरेट ना कर रहा हो वो इंट्रॉनिक सीक्वेंस, तो फिर उस प्रोटीन प्रोडक्ट पे क्या इफेक्ट होगा?
नहीं मैंने आपको कहा डिपेंड्स, ये हम आगे पढ़ेंगे। प्रोटीन्स दे मे गेट ट्रंकेटेड, उस जगह पर आकर वो रुक जाए। ये आपका राइबोसोम इवन हॉल्ट कर जाता है, रुक जाता है ट्रांसलेशन के दौरान, आगे ट्रांसलेशन नहीं कर पाता। अगर कोई इस किस्म की सिचुएशन बन जाए कि जिसमें mRNA में कोई इशू आ जाए, आई डोंट नो, अगर वो आई मीन वो स्टॉप कोडॉन हो सकता है आपका, तो ट्रांसलेशन स्टॉप हो जाएगी वहां पर अगर वो स्टॉप कोडॉन हो गया। अगर वो अल्टरनेट कोडॉन है किसी mRNA सॉरी किसी अमीनो एसिड का, तो दूसरा डिफरेंट अमीनो एसिड उसमें इनकॉर्पोरेट हो जाएगा। तो ये नहीं है कि आई मीन कोड तो सेम होता है, आपके इंट्रॉन इसलिए ट्रांसलेट नहीं हो पाते क्योंकि वो स्प्लाइस हो जाते हैं, सॉरी रिमूव हो जाते हैं। आपके एक्सॉन्स जो हैं दे आर पुट टुगेदर एंड इंट्रॉन्स आर रिमूव्ड। ट्रांसलेशन उनकी इस वजह से नहीं हो पाते, इस वजह से नहीं कि उसके ऊपर कोई डिफरेंट चीज होती है, सिंपली वो मैच्योर mRNA का पार्ट नहीं होते, वो न्यूक्लियस में रह जाते हैं, वो एक्सपोर्ट नहीं हो पाते, और ट्रांसलेशन ने तो साइटोप्लाज्म में होना होता है, तो दे आर एक्सपोर्टेड आउट। जो एक्सॉन्स होते हैं, इंट्रॉन्स आर नॉट एक्सपोर्टेड आउट। अगर इंट्रॉन एक्सपोर्ट हो जाएं दे कंसिस्ट ऑफ द सेम कोडॉन।
जी सर, ठीक है।
और फिर ये डिपेंड करेगा आई मीन वो किस चीज को कोड करना है। अगर वो किसी भी अमीनो एसिड का कोड है तो उस अमीनो एसिड कोड कर देंगे, वो स्टॉप कोडॉन है वहां पर वो मॉलिक्यूल ऐड करके उसको स्टॉप कर देंगे, एंड समथिंग लाइक दिस ना। सो दैट डिपेंड्स अपॉन द कोडॉन, कि वो किस चीज को कोड कर रहा है। आई मीन वो किसी चीज को कोड नहीं करता देन इट्स नॉट अ कोडॉन ना। Okay, so should we proceed now? Hello.
Yes.
Okay, हम ये कुछ चीजें आगे पढ़ेंगे इसमें से, हमारा डेग्रेडेशन में आपको समझ आ जाएंगे कुछ चीजें. So we know that in all the organisms, they need a reliable mechanism to turn their genes on and off, uh, and some of these things we have already discussed as well. But one important determinant is also degradation of the mRNA molecule, that after being produced and exported out into the cytoplasm, this mRNA molecule needs to be degraded. It cannot stay there forever. So it has its own lifespan, and sometimes it gets a signal from somewhere else that this mRNA is not needed anymore, so it should be degraded. So in order to regulate how much a protein is synthesized, it depends upon multiple different things. And one of them is maintaining a level between the production of mRNA and the degradation of the mRNA molecule. So this balance between the production of the mRNA molecules and the degradation of the mRNA molecules would determine the net production of the protein. Kitni protein banni hai. So for this, we need different mechanisms to degrade the mRNA molecules that we will talk about in this lecture. So there are two things that can happen in order to degrade an mRNA molecule. We either need to remove the polyA tail, that's what we call deadenylation, and then an exonucleolysis can take place in 3' to 5' direction from this direction. So first we need to remove the polyA tail, and then a set of exoribonucleases can degrade the mRNA molecule in 5' to 3' direction. And the other mechanism we have on the other side is the decapping of the mRNA molecule. When we want to destroy the mRNA molecule in 5' to 3' direction. So first we need to remove the cap, and then we need some exonucleases which work in 5' to 3' direction. You can also destroy the mRNA molecule by endonucleolytic mechanism, where the mRNA would be degraded in the middle, but that's the general mechanism, and bulk of the mRNA molecules, they are degraded in 3' to 5' fashion, this one. So this is the most common mechanism to destroy the mRNA molecule. There are lots of different, there are quite some differences between the degradation mechanisms. So we will study them one by one, but that's the general mechanism, because these two, the 5' cap and the 3' polyA tail, these two, as we have previously discussed, are the endogenous protective mechanisms for the mRNA molecule. So this cap protects the mRNA molecule from degradation at the 5' end by the exonucleases and polyA tail protects its degradation from the 3' end. So both of these two ends, the 5' end and the 3' end of an mRNA molecule, are protected via a cap and via polyA tail. So if you want to destroy this mRNA molecule, you need to remove the guards at both the ends. So either you start from 3' to 5' direction, then you need to remove the polyA tail, or you start from 5' to 3' direction, then you need to remove the cap. You need to remove the guard so that the exonuclease can destroy the mRNA molecule. So the mRNA, so if anybody has got any questions, you can definitely interrupt me. It's not an issue. So the mRNA decay rates, they can vary in animals and even within the same species, depending upon the type of the mRNA molecule. But the average in mammals, the average lifespan of an mRNA molecule is approximately 8 hours after being exported into the cytoplasm. But it can be pretty fast as well. For example, with C-Fos gene, so it's a proto-oncogene. We have previously discussed proto-oncogenes are those genes which generally do not play the role of being an oncogene. They do not cause cancer, but if something goes wrong with them, if there's some sort of mutation, or if there's overexpression of these molecules, then this may lead to cancer, to the development of cancer. And that's why we call them proto-oncogenes, means they are generally not oncogenes, they do not cause cancer, but if something goes wrong with them, they may lead to the development of cancer. So the lifespan in this case can be pretty short, so approximately 15 minutes, and then after 15 minutes, the mRNA molecule would be destroyed. So they're generally involved in, CFOS genes are involved in proliferation and differentiation of the cells. Most of the oncogenes are basically those which are coming from proliferation or differentiation phenomena in the cells. Or it can be pretty slow. For example, in case of beta globin is part of your hemoglobin. So you have got two globin parts, alpha globin and beta globin. So in case of beta globin, it could be approximately, it can live as long as 17 hours before the mRNA molecule gets destroyed. So it depends upon so as we have seen, it depends upon the type of protein being produced and its role. And it also depends upon we will study this later, it also depends upon the composition of the mRNA molecule. If something goes wrong with the mRNA molecule, if there's some sort of issue that the transcript is not correct, then this may get destroyed as well. So any questions so far?
No, sir.
Okay. So there are lots of different pathways. We would discuss one by one some of the most common pathways. As I have told you that deadenylation-dependent mRNA decay means where deadenylation is mandatory. You definitely need to remove the poly-A tail, otherwise you cannot destroy the mRNA molecule. That's what we call deadenylation-dependent mRNA degradation. And the second one is deadenylation-independent mRNA degradation, means where you do not necessarily need to remove the poly-A tail. You can destroy the mRNA molecule without removing the poly-A tail. So we will talk about the deadenylation-dependent mRNA decay in today's lecture. Then we will talk about the deadenylation-independent mRNA degradation. Then we will talk about the endoribonucleolytic decay. I told you that in addition to these two phenomena, the 3' to 5' degradation or 5' to 3' degradation, which is carried out by the exonucleases, you can also destroy the mRNA from within. That's what we call endonucleolytic degradation of the mRNA. So we have deadenylation-dependent mRNA decay, deadenylation-independent mRNA decay. We will talk about the endoribonucleolytic decay. We will then we have fourth type where we say nonsense mediated decay or non-stop and no-go decay. That's what Sirat asked about. So we will talk about this question in details in this category. So nonsense mediated decay means where you cannot proceed with it. It's a nonsense mutation. And then we have non-stop means there's no stop codon, something like this. So we will discuss the details of these things. So that these would be it would depend upon the amino acid sequence of the mRNA molecule. And then we have AU-rich elements and their binding proteins. That's the fifth phenomenon. We have discussed that poly-A tail also when we were talking about the addition of poly-A tail, if you remember. Rather, we were talking about alternative polyadenylation, I think. Yeah, here, poly-A tail. We talked about, I don't know, here, alternative polyadenylation. So I told you that this can also determine the lifespan of the mRNA molecule. So AU-rich regions or AU-rich animals, AU-rich elements are generally found in this 3' UTR. So this would also determine the lifespan of the mRNA molecule. So that's what we will study later. So that would be the last type of mRNA degradation. So we will start with the first one today, the deadenylation-dependent mRNA decay. And then step by step, one by one, we will also talk about the other categories. So let's start with the first one, the deadenylation-dependent mRNA decay. So as the name indicates, it is dependent upon deadenylation, means you cannot destroy this mRNA molecule without destroying the poly A tail first. So these are two integral stability determinants, the 5' cap and the 3' poly A tail. Because both of these two, they interact with the cytoplasmic proteins in order to protect the mRNA molecule. We know that poly A tail is bound by the poly A binding proteins, PABC and PABN1 in the nucleus and PABC in the cytoplasm. N stands for nucleus and Z stands for cytoplasm. So they remain bound to the poly A tail, and there are lots of other proteins which also remain bound to the 5' cap. And interaction of these proteins with the cytoplasmic interaction of these two ends, the 5' cap and the poly A tail, with the proteins in the cytoplasm protects both of these two ends from exonucleases and provides stability to the mRNA molecule. So in order to initiate decay, either one of these two structures needs to be destroyed, either the 5' cap or the poly A tail. So in case of deadenylation-dependent mRNA decay, so do not confuse it with 3' to 5' or 5' to 3' decay, because that's the direction of the decay. Even 5' to 3' decay can be dependent upon deadenylation. You want to proceed, you want to destroy the mRNA molecule from this side, you want to remove the cap. But if this is the deadenylation-dependent mRNA decay, you would definitely need to remove the poly A tail, and then degradation would start from 5' towards the 3' end. But if the degradation of the mRNA is 3' to 5', then you need to remove the poly A tail first, and then the mRNA molecule would be destroyed. And in both the cases, we first need to remove the poly A tail. That's what we call deadenylation-dependent mRNA decay. So what generally happens that because I told you whether you want to go 3' to 5' end or you want to go 5' to 3' end, you first need to remove this poly A tail. This is what we call deadenylation-dependent mRNA decay. So this is your mRNA molecule with 5' and 3' UTRs, ORF in the middle, and this is the 7-methylguanosine, the cap of the mRNA molecule, and that's the poly A tail. Because this is deadenylation-dependent mRNA decay, the first thing you need to do is to remove the poly A tail. So poly A tail is removed by certain enzymes. We will study them later, how this phenomenon takes place. We will study both of these two things one by one. So the first thing you need to do is to remove the poly A tail, and then it can go into either direction, either to 5' to 3' decay or 3' to 5' decay. Now we will study both of these two phenomena one by one. So first the 3' to 5' decay, and then 5' to 3' decay. So what you need to understand till now is that you need to remove the poly A tail first. Clear to everyone?
Yes, sir.
Okay.
Yes, sir.
Okay. So we will study this thing first, and then this step, and then that would be the third step, 5' to 3'. But in both the cases, whether it is 3' to 5' or 5' to 3', this is our deadenylation-dependent decay. Means we need to remove the poly A tail first. So it is not yet clear how this phenomenon is triggered, the degradation of the mRNA molecule, but there are lots of deadenylase enzymes which are involved in degradation. So this is a picture, this is a description of deadenylation in Saccharomyces cerevisiae. And you can see this is the normal mRNA molecule, which is capped at the 5' end, and it has a poly A tail at the 3' end. And in order to remove the poly A tail, we need some nucleases, what we call poly A binding protein dependent polyanucleases. So we know that this poly A tail remains bound with certain proteins, and generally these proteins are poly A binding proteins. So if it is in the cytoplasm, so that would be PABC, poly A binding protein, which is inside the cytoplasm. So these enzymes which would bind to these proteins which are associated with the poly A tail. So these enzymes would bind to the proteins which are associated with this poly A tail. So you would have certain proteins here, and these enzymes would recognize those proteins and they will bind to the poly A tail. That's why we call them poly A binding protein dependent poly A nucleases. So poly A nuclease means it removes the poly A tail, and PABP dependent means it is dependent upon poly A binding protein. So it cannot bind to a naked poly A tail. So in case of any mature mRNA molecule, this remains associated with the poly A binding proteins. So whether it is CCR4, so CCR stands for chemokine receptor, and so that's one example. This is the name of the deadenylase molecule, or there's another one which is known as PAN. PAN stands for poly A specific ribonuclease. These are also, this is another example of PAN2, PAN3. So all these names, you do not need to certainly remember them, but all of them are, they are the names of the deadenylases. And these deadenylase molecules, whether it is CCR4 not, it is PAN or it's PAN2, PAN3, they are dependent upon, आप एक second hold करें. अस्सलाम वालेकुम. वालेकुम. जब मैं क्लास में हूँ, मैं फ्री होकर कॉल करूँ. जी सही है, ठीक है. अच्छा पार्टी है, बस ठीक है, मैं बात करता हूँ. बस ठीक है ज़बरदस्त. हाँ ठीक है, ओके. हेलो, आवाज़ आ रही है आप लोगों को?
जी सर, आ रही.
सॉरी फॉर डिस्टर्बेंस, एक अर्जेंट कॉल आ गई थी और बंदा बार बार कॉल कर रहा था. So apologies for that. So we were talking about the deadenylation. So means these are the names of these different enzymes you might come across, but all of them are basically poly A binding protein dependent poly A nucleases, which are involved in the trimming of the poly A tails. So after this has been trimmed down, the poly A tail is now removed, and we want to carry out 3' to 5' decay. Then that would be done by the exosome. Exosome is a 10 to 12 subunit complex where there are generally 6 of them are proteins, and all of these proteins, they have got these endonuclease or exonuclease or helicase activity, means they have the ability to destroy the nucleotides at the 3' end, and they're put together, so they're individual proteins, 6 of them, and they constitute a complex. So all of them are basically they're exonucleases or helicases, means they have the potential to destroy the mRNA molecule, but they work at the 3' end, and they work in the form of a complex. And this entire complex of different proteins, we call it an exosome. So 3' to 5' degradation is carried out by exosome. So after the mRNA molecule has been destroyed, then we are left with this small oligonucleotide and the cap at the 5' end, and that is destroyed by another scavenger enzyme, which is known as the DCPS. So DCP stands for decapping enzyme, and S stands for scavenger. So this, as you can see, this is basically the cap here, which is denoted. So this is simply indicating that this enzyme has the potential to remove the cap, and that this one indicates that this is the active site of the enzyme which can remove the cap. So the small number of nucleotides, the remaining nucleotides, and this cap, they are destroyed by the DCPS at the end. So this is how 3' to 5' decay is carried out. First, you need to remove the poly A tail by certain deadenylases, which remove the poly A tail, and then a complex of proteins, what we call exosome, where there are roughly 6 proteins, each of them with an RNA destroying ability. This exosome complex, this attacks the 3' end and starts destroying the mRNA molecule in 3' to 5' direction, and last few nucleotides are left undestroyed, and basically because they need to be destroyed by the scavenger enzyme, which in this case is the DCPS. and this DCPS would destroy the remaining oligomer as well as the cap. And this is how mRNA would be destroyed in 3' to 5' fashion. Any questions so far?
No, sir.
It's clear to everyone?
Easy है.
Easy, it's very easy. Sara easy.
Yes, sir.
So then we have the second option, that because this is probably this is deadenylation-dependent degradation. So now we are moving towards the other direction, which is 5' to 3' decay. And we first destroy the poly A tail by the same deadenylases, and then rather than proceeding in 3' to 5' direction, in this particular case, the mRNA is destroyed in 5' to 3' direction. So in order to destroy it in 5' to 3' direction, what is needed after removal of the poly A tail, we need a signal that should trigger the degradation from the 5' end. So that is carried out by the binding of LSM proteins. So LSM stands for, so basically these are SM proteins or like SM proteins. L basically stands for like SM proteins. SM proteins are, there's an autoimmune disorder basically. So these proteins are generally involved in lots of mRNA processing. Generally, they're involved in processing mRNA molecules. And the name is derived from an autoimmune disorder, what we call systemic lupus erythematosus. This is the name of the disease. And basically, they were these proteins can be detected by the same antibodies. That's why we call them like SM. That they are in systemic lupus erythematosus. We have these proteins, SM proteins, systemic lupus erythematosus proteins, and these proteins can be, because they could be detected by the same antibodies, that's why they were named as LSM, means like SM proteins. And SM proteins are those which are found in this autoimmune disorder. But generally, this is a family of proteins, and the job of these LSM proteins is to metabolize the mRNA molecules, and they generally work by constituting a haptamer. Haptamer means there are a total of 7 proteins: LSM1, LSM2, 3, 4. So we have 10 minutes left. So we have LSM1, 2, 3, 4, 5, 6, and 7. So these are 7 subunits. There are 7 proteins which bind together in order to constitute this complex. This complex then binds to the 3' UTR. So this 3' UTR contains signal for the binding of LSM proteins. So this complex of LSM proteins would bind to the 3' UTR, and this would recruit an mRNA decay factor, which is known as PAT-L1. Decay factor means this is a factor, this is a protein which is involved in mRNA decay. So that would trigger the mRNA decay, but it would trigger mRNA decay at the 5' end rather than initiating at the 3' end. So first, deadenylation, and then LSM proteins, 7 of them, they constitute a haptamer, and they bind to these monomers constitute. So 7 of them we call it a haptamer, because there are 7 proteins or 7 monomers. They constitute this complex, and they bind to the 3' UTR, and they recruit PAT-L1, which is an mRNA decay factor. And what does PAT-L1 do? So here we see this is LSM1. So this recruits PAT-L1, and then PAT-L1 recruits another dimer of this, another decapping enzyme. So this decapping enzyme in this particular example, this is an example from Saccharomyces cerevisiae. So this is a complex of it's basically a dimer. It means there are 2 proteins, DCP1 and DCP2. So DCP stands for decapping enzyme, DCP1 and DCP2. And here DCP2 has got this this domain which destroys the cap. So M7G means that 7-methylguanosine. So it has got this domain which is known as MUT-T, M-U-T, and then capital T. MUT-T is the name of the domain. So this DCP2 contains this catalytic domain which destroys the 5' cap. And this DCP1, DCP2 complex is recruited by PAT-L1. So that's why PAT-L1 is known as mRNA decay factor. After binding to the LSM1, the Patal1 would bind here, and this would recruit DCP1, DCP2 complex, so that this complex can remove the cap from the 5' end. So after this cap is removed from the 5' end, because this was a decapping enzyme, it didn't have anything else to do. We have the entire mRNA molecule still intact except the poly-A tail, which has already been removed. So we need to destroy it in 5' to 3' direction. So in order to do this, we need an exonuclease. So this is not an exonuclease. This was a decapping enzyme. The job of this enzyme was just to remove the cap. So it removes the cap, and after the cap is removed, the mRNA molecule is naked at the 5' end. And then another exonuclease, which is known as XRN1. So XRN basically stands for exoribonuclease. So XRN1 is recruited, and XRN1 starts destroying the mRNA molecule in a 5' to 3' fashion. And this is how ultimately this mRNA molecule is removed. So it doesn't need to do anything at the 3' end, because already poly-A tail has been destroyed by the deadenylase, like CCR4 and PON or PAN2, PAN3, whatever. These are different enzymes because the poly-A tail has already been removed. So we do not need to do anything at the 3' end. So it keeps moving in 5' to 3' direction, and it destroys the mRNA molecule. So that's it for today. I think it is sufficient. So what we learned today is that the mRNA molecules need to be degraded after a certain time because they're not needed anymore. And they have different lifespans, starting all the way from 15 minutes to 17 hours. And then we discussed there are different pathways to destroy mRNA molecules, and we just discussed the first one, which was deadenylation-dependent mRNA decay. And we studied that in this case, the mRNA is protected at both the ends. And so whether we proceed in 3' to 5' or 5' to 3' direction, we first need to remove the poly-A tail. And then we talked about the two phenomena, pathways, 3' to 5' and 5' to 3'. So in 3' to 5', after removal of the tail, we need an exosome complex which destroys the mRNA molecule, and the cap is destroyed by DCPS at the end. While in case of 5' to 3' degradation after removal of the poly-A tail, LSM proteins bind to the constitutive haptomer, which binds at the 3' UTR. It recruits Patal1, which is an mRNA decay factor. This Patal1 then recruits DCP1, DCP2 complex, which removes the cap. And after the cap is removed, XRN1 starts destroying the mRNA molecule from 5' end towards the 3' end. This is how polyadenylation-dependent mRNA decay is carried out. So thank you very much for your attention. So if you have any questions now.
सर ये जो 5' to 3' and 3' to 5' डायरेक्शनैलिटी ये कौन डिटरमिन करता है, mRNA की नेचर या उस पे बाउंड प्रोटीन्स?
Generally mRNA key sequence. I mean, we have these things already coded in the mRNA molecule. We will study some of them later. But like I told you, what triggers it is not exactly known.
जी सर.
What triggers it? Whether it is the sequence itself inside the amino acid. So what would bind to the mRNA molecule, that depends upon the composition of the mRNA molecule. So we will study this later, that different types of mRNA molecules have different degradation mechanisms. While most of them are destroyed in this fashion, most of them. But how is this triggered? That okay, this is not needed anymore and it should be destroyed, that is still debatable, or I mean, there could be some research papers, there could be an update, I don't know about it. But this is the mechanism where bulk of the mRNA molecule is degraded. That's poly-A dependent deadenylation. But what exactly makes these differences? There are certain things, but I mean, we cannot perhaps talk about all of them. We will look at the few examples in our following classes.
Yes, okay.
Yeah. So any other questions?